Hi guys, in this video in biology we're taking a look at mitosis. So this is a process of the cell cycle that we've talked about. So the cell spends most of its time in interphase and then it will go into cell division and mitosis. And mitosis is subdivided into PMAT, Prophase, Metaphase, Anaphase, and Telophase. So let's go ahead and dive into each one of these particular phases and what is happening in the cell at each phase. So prophase, the P. So in this phase, the chromatin fibers become more tightly coiled, so they're condensing into more discrete chromosomes. So the DNA material is all getting packaged and getting ready to be moved and separated. And then the nuclei will begin to dissolve, and the duplicated chromosomes will appear as two identical sister chromatids. So those identical sister chromatins, what that basically means is that the DNA has completely copied, and now you have two identical copies of the DNA ready to be split up into two identical cells. And then on top of that, if you notice on the outside, you have the mitotic spindle. So these are the centrosomes on the outside edges that are slowly making their way to the polar ends of the cell. And they're beginning to form at this point, and the microtubules are also beginning to extend out, ready to connect with each of the chromosomes. Now in between metaphase and prophase, you have a prometaphase stage. This is where the nuclear envelope will begin to fragment and dissipate, and the microtubules again will further extend into the nucleolus now. It will now break through that nucleolus and grab onto the sister chromatids, each one of the chromosomes. Now when you look at the chromosomes, you'll notice that in the middle they have that little protein called the kinetochore. And that protein is the structure where the microtubules will then connect with the sister chromatins and will begin the process of separating them. And then soon after prophase or prometaphase, you have the longest stage of mitosis, which is metaphase. This is about a 20-minute long process where the centrosomes are now on opposite poles, and the microtubules have grabbed onto the kinetochores, and they will now pull the chromosomes equidistant to each one of the centrosomes. So essentially they put them on what's called the metaphase plate. That metaphase plate is an imaginary line which splits the cell up in halves, two equal halves. The chromosomes are now getting aligned on that metaphase plate. And now we go into anaphase. Anaphase is the shortest stage of mitosis, only lasting a couple of minutes. Now the two chromosomes that they're being liberated from each other, they will begin to move toward the opposite ends of the cell, being pulled by their kinetochores and microtubules. So those microtubules are now beginning to shorten, and they're getting pulled closer to the centrosomes, and those centrosomes are pulling the chromosomes apart. Anaphase. Anaphase, the letter A, they're pulling away from the center. And then metaphase, think of them being pulled towards the middle. So anaphase are being pulled away towards the poles. And then finally we end up with telophase. Telophase, you have two daughter nuclei that are now beginning to form two brand new cells. The nuclear envelopes are beginning to reappear, and they're starting to form around the chromosomes that are now breaking up and becoming less condensed. So imagine now you moved into your new place and you're now starting to unpack. So mitosis at this point, instead of having one cell with one nucleus, now you have two identical cells with two identical nuclei having identical forms of DNA. And then last but not least, we have cytokinesis. This is the actual division of the cell where it develops what's called a cleavage furrow. So they start kind of pinching at the middle, and they pinch so much so that they essentially meet, dividing the cells, forming two brand new cells where we had one before. And that pretty much does it for mitosis, PMAT, prophase, metaphase, anaphase, and telophase.